Thank you for kind introduction. It's such a pleasure for me to represent Sean to join this symposium and discuss cancer microbiome. Our lab focuses on the intratumor bacteria and host cancer cell interaction. So today, I'm going to show you a very interesting story about how intratumor bacterial invasion influence the breast cancer cell metastases. If I sound a little bit nervous, please forgive me because I'm so excited to be here as a postdoc. So first, let's think about cancer. Cancer is a very complex ecosystem and is composed with many cell types, including the cancer cells, endothelial cells, fibroblasts, and all kinds of immune cells. And all these contribute to the heterogeneity of tumor microenvironment. And interestingly, the tumor microenvironment has been characterized as hepatic, necrotic, and immunosuppressive, and it's proposed to be favorable for bacterial growth. However, it is very challenging to detect bacteria in tissue samples because of its low abundance. So it's highly debatable whether these bacteria are coming from the environment or from the tumor tissue samples. Actually, the researchers in this field try to figure out these questions, and in 2020, Revius Dressman Lab optimized the bacteria DNA detection methods and claimed that intratumor bacteria are broadly present in tumor tissues, including those which were thought to be sterile, like brain, bone, and breast tissues. And our lab also improved the bacteria DNA characterization methods in tissue samples, and we reported that in the murine spontaneous PMT breast cancer model, there are culturable bacteria in breast tumor tissues, and the most abundant bacteria are coming from the Staphylococcus, Streptococcus, Enterococcus, and Lactobacillus, which are commensals for the host. And actually, these intratumor bacteria are not just passengers. Both correlative and functional studies review the key roles of intratumor bacteria in drug resistance, cancer initiation, metastasis, and immunoregulation. And interestingly, the engineered intratumor bacteria showed promising efficacy in cancer treatment. And in addition to these diverse physiological functions of intratumor bacteria, researchers in this field study to appreciate that the bacteria can not only live in tumor tissues, but also inside the tumor cells. The first report in 1965 showed bacteria inside tumor cells in a spontaneous new tumor and proposed that this kind of intratumor bacteria can be a causing agent for tumor formation. And several years ago, Revius Dressman Lab, using correlative light and electron microscopy, found that the bacteria are closely related to the intracellular, related to the host cell nucleus. And our group also observed the intracellular bacteria in a spontaneous murine breast tumor. So it is very interesting to think why the bacteria localize in the cytosol and what's the physiological meaning of it. In our previous work, we're using antibiotics to deplete intratumor bacteria specifically and found one of the major physiological functions of intratumor bacteria is to promote long metastasis colonization. And the mechanism is that the bacteria inside tumor cells can reorganize the cancer cell cytoskeleton and help cancer cell resistance to fluid shear stress. So the tumor cell with bacteria can survive and colonize better in the distant organ. However, the metastasis is a multi-step process. The mechanical stress takes in action during the very early stage of metastasis. For example, here, if we inject PMT ruciferous tumor cells back into mice through the tail vein, and we can observe drastic reduction of cancer signals in the lung, no matter in FVB immunocompetent mice and immunocompromised mice. And however, in the later stage, only the immunocompromised mice can allow cancer cells to efficient relapse. So this indicates that there is a second wave of surveillance by the immune system to kill the rest of metastatic cancer cells. In light of our previous discovery that circulating tumor cells, particularly circulating tumor cluster cells, contains amounts of bacteria. So our scientific question here is, when the circulating tumor cells carry bacteria to the distant organ, do these bacteria remain function during immune surveillance in the later stage of metastasis? So to answer these questions, we established the in vitro bacteria and the tumor organoid co-culture system to mimic circulating tumor cells containing bacteria. And here we use Staphylococcus dialysis, which is a predominant bacteria in primary tumor tissues, and we add penicillin, streptomycin antibiotics to the co-culture system. These two kinds of antibiotics are cell impermeable, so it can kill the extracellular bacteria efficiently. So you see from the 3D co-culture reconstructed view of tumor organoids, we can observe that most bacteria are inside tumor cells. And further EM analysis confirmed the existence of intracellular bacteria and showed high efficiency of intracellular bacteria invasion. So this co-culture is a very nice system to study the intracellular bacteria. So using this co-culture system, we ask, after the first wave of mechanical stress, whether the bacteria still play function in the later stage of metastasis. So we injected POMT luciferase organoids with bacteria back into mice through the tail vein, and after tumor organoids home into the lung, we treated one group with doxycycline, which is a cell-permeable antibiotics, can specifically deplete intracellular bacteria. Very interesting, as we checked lung signals in the cancer signals in the lung, and we found the presence of bacteria can efficiently for the cancer relapse, while the doxycycline to deplete intratumor bacteria inhibit cancer recurrence. And the cancer recurrence inhibition effect is not due to direct cancer killing by the doxycycline, because when we did the same experiment in the immunocompromised NPSG mice, the inhibition phenomena was completely disappeared. So this suggests that doxycycline-mediated relapse inhibition is immune-dependent and probably due to the depletion of intratumor bacteria. So we asked which kind of immune cells respond to the intratumor bacteria firstly. So we injected the tumor cell and the tumor cell with bacteria back into mice through the tail vein and profiled long immune infiltration. And we found that neutrophil is the major corresponding cell to the intratumor bacteria. And other immune cells didn't show the significant change. And the tail vein injection model only mimics some steps of metastasis. So we also tested under physiological condition whether elimination of intratumor bacteria can reduce the neutrophil population. So we performed the experiments on germ-free mouse model to avoid gut bacteria interference. So you see here, the bacteria containing tumor group showed a strongly increase of neutrophil infiltration, while antibiotics to deplete intratumor bacteria efficiently decrease it. The neutrophil is the innate immune response cells, so it is reasonable for neutrophil increase under the existence of bacteria. But the question here is, when the bacteria are within the tumor cells, how neutrophils sense and respond to the bacteria? Actually, neutrophils are heterogeneous in tumor microenvironment, and both anti-tumor state of neutrophil and the pro-tumor state of neutrophils have been described in literatures. So here, our hypothesis is that intracellular bacteria can convert neutrophils into a pro-tumor state and reshape the immunosuppressive microenvironment and promote long metastasis recurrence. To test our hypothesis, we set up 4 groups: PBS, bacteria-free tumor, out bacteria tumor, in bacterial tumor group to do single-cell RNA-seq of long immune cells after 12-week injection. And very interestingly, when we analyzed the neutrophil state, we found extracellular bacteria promote neutrophil anti-tumor score while decrease neutrophil pro-tumor score. And on the contrary, intracellular bacteria can enhance pro-tumor neutrophil while suppress anti-tumor neutrophil. Now, the question here is how bacteria-invaded tumor cells determine the neutrophil state. We went back to our previous single-cell analysis of bacteria-invaded tumor cells and asked how host cell respond to the bacteria invasion. So after the single-cell analysis, we found that except the bacterial infection and NF-κB related pathways, IL-17 pathway is the most enriched pathway after bacterial invasion. We then checked all the ligands of IL-17 families, and the most frequent studied IL-17A and IL-17F don't express in the tumor cells and the tumor cell with bacteria. It is IL-17B upregulated in the tumor cell with bacteria. And IL-17B is very different from other IL-17s because it is specifically expressed in epithelial cells, not immune cells. So we wondered whether IL-17B can mimic intracellular bacterial function to mediate neutrophil reprogramming. So we treat neutrophils with IL-17 protein directly, and we found that neutrophils were reprogrammed to the reprogrammed to a state that significantly suppressed T-cell activation by testing the IFN gamma expression. And more interesting, if we overexpress IL-17B in tumor cells, and we can find that it is sufficient for tumor cells to escape immune surveillance and give rise to efficient relapse. And all these experiments suggest that the extracellular bacteria all these experiments suggest that intracellular bacteria may reprogram neutrophil state or prevent metastasis through IL-17B. But the question here is whether extracellular bacteria can turn on IL-17B or not. Well, to answer these questions, we designed two experiments. The first one is transwell experiment, which allow us to separate the intratumor bacteria and the host cells. So you see here, only the bacteria invaded the tumor cells upregulate IL-17B, while when the bacteria is spatially separate with the host cells, the IL-17B expression don't change. And another experiment is cytokinase D treatment. Cytokinase D can depolymerize the actin filament and inhibits endocytosis and the invasion of tumor bacteria. So after the cytokinase D treatment, the IL-17B upregulation by bacterial invasion was strongly abolished. So these two experiments tell us that IL-17B upregulation is intracellular bacteria-specific response. So we wondered whether this dedicated mechanism holds true in the human system. To answer this question, actually we have been collecting the human breast cancer samples for 5 years and found that the bacteria truly exist in the human tissues by imaging. And we also try to isolate the human breast cancer specific Staphylococcus, and the statistics results show that in human breast tumor tissues, the most abundant Staphylococcus is epidermidis. It's not different from the Staphylococcus zealosus in the mouse samples. We then use the isolate human breast cancer specific epidermidis to co-culture with the human breast cancer cell lines and do the bulk RNA-seq of tumor cell with or without bacteria. So you see here the tumor cell with bacteria invaded show different gene signature and enriched IL-17 signaling pathways. All this is highly consistent to our mouse data. And in addition, we also isolate neutrophils from healthy donor, and we found that the live bacteria invaded tumor cells can reprogram neutrophil into suppressive state, which highly expressed as 100A8A9, PD-L1, 6L2, PTGS, while the dead bacteria, which we presumably cannot inside the tumor cells, cannot do that. So next question is how bacterial invasion of cancer cells influence breast cancer patient survival. Well, to answer these questions, we assembled Staphylococcus epidermidis invaded gene signatures from our BOC-RNC data and stratified human patients into high-level and low-level gene expression of bacterial invasion. So you see here, the worst prognoses are significantly correlate with the bacterial invasion gene signature. And similarly, the neutrophil response to Staphylococcus epidermidis tumor cells and IL-17 pathway are all significantly prognostic. So this data show that bacterial invasion is actually can be a cancer prognosis. Can be a biomarker for worse prognosis for human cancer patients. Well, the last question is, can targeting intracellular bacteria improve post-surgery cancer outcomes? To answer these questions, we set up a model to mimic clinical condition. First, we injected bacteria-containing tumor cells, and when the tumor grew into a certain size, we did a surgery to remove the primary tumor, and then treated one group with doxycycline to deplete intratumor bacteria. And very interestingly, after surgery, the doxycycline treatment can still have function and strongly suppress the long metastasis recurrence. And the recurrence rate was reduced from 80% to 20%. And according to our single-cell analysis, the extracellular bacteria play opposite roles from intracellular bacteria in immune regulation. So here we decided to use the bacterial cell wall component, PGN and ILPS, to represent extracellular bacterial effects. After the surgery, we treated the mice with PGN and ILPS, and we found that extracellular bacterial component treatment can strongly decrease long metastasis recurrence. So all these experiments show that appropriate management intratumor bacteria can benefit the cancer patient after surgery and prevent cancer metastasis recurrence. Finally, to summarize our work, we found that in addition to mechanical stress, the intratumor bacteria are important for cancer cells to cope with immune in cancer metastasis relapse. And interestingly, the extracellular and intracellular bacteria play distinct roles in shaping the immune microenvironment. Specifically, intracellular bacteria turn on IL-17B to introduce neutrophil suppressive state. And finally, the therapeutic application should consider bacterial state and antibiotics properties. Last but not least, I really want to thank my supervisor, Dr. Shang Cai's mentorship during my PhD journey, and I want to thank all the lab members and the collaborators, and I want to thank Westlake University Bioinformatics Research Facilities and the lab animal resource and the biorender illustration. And finally, I really want to thank the opportunity to join this meeting and share our recent work to all of you. Thank you for your listening.